Okay, yeah, welcome everyone for the Lunch and Learn webinar. And this is the webinar series for the GREENIN Micro Network Plus. This is webinar number 49, but I believe that this is around the 6th or 7th webinar for the GREEN network. So it's a legacy of Reclaim Network Plus, which we were continuing. That was focused more around the outdoor greening, and this webinar series is focused around the indoor greening. So just to say very quickly, the Green Micro Network is funded by the UKRI, especially from the EPSRC, and I have a pleasure of leading it from the Global Centre for Clean Air Research with colleagues from the UKCH, Bath, Oxford, Cranfield, and York. So next slide, please. So the this webinar series usually, it happens. Can we move to the next slide, please? It usually happens every Wednesday. So that's the first month of the, so first Wednesday of the month, and between 12:30 and 13:30. So somehow I can still see the same slide there. Did you move that, Sarvan, the slide?
Yeah, it's for the previous webinar number 48. I move it, so maybe I can go back to the second slide, which is the core management.
Can you go to the second slide, please?
Yeah, it's the core management team.
Yep. So yeah, that's the team basically who has who comes from different institutions, but they also got a very specific, the expertise are linked to the particular topic area. So I'm not going to go into detail. So next slide, please. So number 3 now.
Yes, it is number 3.
Okay, so the previous webinar, you can see we got 2 fantastic speakers coming from the UK and Amsterdam. And so this webinar series basically, as you said, we usually have 2 speakers, try to diversify them with the academic and non-academic, but sometimes we got the experts, from an individual sector. And all these webinars are recorded. So you can see them on our YouTube channel. So basically you could watch actually the previous webinars if you wanted to have a look on the diversity of the topics which are covered there. So we'll paste the link into the chat box. Next slide, please. So yeah, just to announce actually the winners for the sandpit. So that's a really good opportunity for us to we run the competition for the funding, and we were allowed to fund 4 projects. We got a very extensive the interest, and had a number of applications which came through. So there were 4 of these the winners for the project. So Dr. Akash Biswal, Dr. Tracy Epton, Dr. John Kaluit Coltit, and Dr. Hausen. So congratulations to all of our winners, and you will see actually a lot more interesting research coming in this particular topic the area there. So next slide, please. So just to also say, I think we just possibly making use of you being there. So possibly we are running a survey which is basically how you can do the DIY work around your household, especially using the greening. So if you get an opportunity to complete the survey, we would be very grateful for you. So we'll put a link in the chat box again for you. Next slide, please. So today, so we got 2 fantastic speakers from Australia. So they have been very generous to give their time. They give up their evening for us. But you might have already seen their fantastic work, which they have done, over the years. And it's a real pleasure actually to have Dr. Fraser Torpy and Dr. Peter Irga. You might have seen a lot of their work today. I mean, in the past, but they will be speaking about their particular topics. So Dr. Fraser Torpy will be talking about the potted plants and indoor pollutant removal, what 4 decades of research tell us, and we'll be hearing from Dr. Peter Irga about the active botanical biofilter systems, which is really something which is emerging these days. So these are really important topics, and we have been lucky to have both of them. So before any further ado, so I'm going to invite our the first speaker. But before I do that, just to let you know that these webinars are recorded, and you will have an opportunity to share or have a look at the webinar later on if there are any particular aspect of the work which really interests you. The other thing is that if you have any questions, please do use Q&A chat box, which is on your screen, and put your name and your question for our speakers. So each of the speaker will speak for 20 minutes, and then after that we will take all your questions, and I'll read out to our speakers to answer them. So in the meantime, you bring your slides up. So let me introduce formally Dr. Fraser Torpy, who is an associate professor and the director of plants and environmental quality research group at the University of Technology, Sydney, where he leads one of the world's foremost research programs on plants and indoor air quality. His landmark publications, among the first in the world to quantify the removal of volatile organic compounds by indoor plants, have been top-cited, help establish phytoremediation as a recognized field. And Dr. The Torpy's work, current research focuses on potted plant systems and on translating laboratory findings into real-world built environment applications. It's a real pleasure to have you, Dr. Torpy. So yeah, without any further ado, over to you.
Thanks so much, Prakash, and it's an honour to be here. Absolutely, always happy to talk to our wonderful UK colleagues. Thanks for their excellent introduction. That saved me a lot of time. The only thing I would like to add to that is that we've been at this for quite a long time now. Our group started in this field in the late 90s, and we've specialised in empirical studies. So we really focus on measuring things in the lab and in the field rather than modelling. So I hope what we've learnt from that 30 odd years is of value if I communicate it to you. Thank you again for the introduction. I think given that I know this audience is quite knowledgeable in our field, I don't need to go into too much detail, and you probably realise this already. The field is a growth field. When I started working in this field in about 2000, we did our first study. It was quite a significant study, and we tried to get it published, and we sent it to 5 journals and it got rejected. No journal would accept the idea that plants are useful at removing indoor air pollution. Now we eventually got that paper published, our first one. It's got over 400 citations. Things have changed. People have recognised the value in what we do, and the field is in a stage of exponential growth, both in Australia, UK, all around the world. Part of the reason for that is, we were talking about a minute ago about, is that the fields move beyond the traditional horticultural science now. We now have a significant number of engineers working in the field, as well as medical people for the health effects, built environment people for the integration into the urban environment, and so on. And with that spread of fields has come a rapid increase in publications. This is a growth area, and we see no signs of that slowing. Internationally, this is a week ago, this data, covering the whole period from 2015 to 2026, looking at who's doing the work. Of course, China is leading. China puts out a great deal of research. They have a lot of money floating around. So naturally, they put out more papers in that period than anyone else. Well, similarly with the Americans, they had a lot of money, and they've used that for research purposes. After that, South Korea, Dr. Kim's group, for those of Peter's laughing at me. Our friend Dr. KJ Kim from South Korea, who's got a fantastic research group, once again, really well funded down there in Jeonju in South Korea. Where next? Japan, Thailand, India. Great to see the UK in 8 That's Tiana's group, I think, is the engine room for that productivity. You will notice, however, though, that number of papers doesn't equal number of citations. Us and the UK are clearly producing the better work in that space. Looking forward to more competition from the UK. Let's go. Catch up to us. The fact that plants can remove air pollution isn't a secret anymore. And to this audience, I don't need to explain how it works. Everywhere around the world, in both the indoor and particularly the outdoor environment, people are putting in trees explicitly for the purpose of mitigating air pollution, particularly challenged areas, India, Pakistan, that have got notably bad air quality, but also even in London. No secrets. In the indoor space, as once again you probably all know, this idea of plants mitigating poor air quality comes from the old NASA studies of Bill Wolverton, who grew their model spacecraft to try and work on the sustainability of life in sealed environments in outer space. So they grew plants for oxygen and food, and as a side effect, the air pollutants in their model spacecraft were extremely low. And of course, in the 80s, the air quality or the pollutants emitted from electronics and plastics and so on were atrocious. But yet in those model spacecraft, they had excellent air quality, and Wolverton had the stroke of genius that started our whole field in tying that back to the activity of the plants. That was a long time ago. It's now 43 years later from Wolverton's work. Geez, that went quickly. We can now, from our end in the empirical space, we can say with confidence that pretty much any potted plant can remove pretty much any concentration of pretty much any volatile organic compound. I think we'd be confident in saying that. No one's tested PFAS yet. It's not really volatile. There's a handful of things that haven't been tested, but in general, that statement is going to hold true. Any plant, any concentration, any pollutant. Concentrations all the way from a handful of parts per billion up to 1,000 parts per million is the highest concentration we've tried, which is an outrageous concentration of volatile organics. And yet the plant still, one single plant in a chamber pulled that down in 24 hours. The capacity here is immense and broadly untested. We can also confidently say, and the literature is a little confusing over this because a lot of, particularly the group from Thailand, do a lot of stuff on plant leaves mitigating air pollutants. But looking at the broad scope of how this process actually works for volatile organics, it is the substrate that does most of the work. So it is the potting mix and the bacteria therein that pull down most pollutants. The plants do, some pollutants are plant mediated. Highly hydrophobic, small molecular weight pollutants like formaldehyde are plant mediated. But all the hydrocarbons are remediated by bacteria growing in the substrate of plants. I say that with absolute confidence now. The process is consortial, it's not trivial. You can't go and identify one pseudomonad that does all of the hydrocarbons. There's a consortium of bacteria in the substrate of plants that perform these actions, and it's a complex process that is poorly resolved. There is inadequate research in that space to work out which sequence or which chronosequence of bacteria actually do this. We also know the plant's critical for the process. So whilst it's a substrate doing the work, the plant can't be removed. We've done the experiment. We're trying to test just simple substrate removing volatile organics. It works for a little while, and then eventually the bacteria lose the sources of nutrients that come from the plant and they stop working. Facts. I'm not supposed to say facts in science, but them's facts. Now here's a graph of an example. Here we've got the bottom line. You see my cursor? Yep, thanks. Bottom line's plant in soil. We got chamber. We've got a very high concentration of benzene in the chamber. I blame Peter Irga for that, the first author on this paper. And as you can see, the plant removes that. This is initial dose, 6 days, removes that very high dose of benzene. The second line is the plant in hydroponics. It's slower because the hydroponics have a lower bacterial density in the substrate. The third line with the upside down triangles is the substrate by itself. It works well, but it's slower than the soil substrate with a plant growing in it. And finally, the top, there's hydroculture medium only because it's got very low bacterial density, so it removes substrates very slowly. This is a plant in a chamber with pollutants, and the data is measuring the decay of those pollutants. There are hundreds of papers that do this. They're still coming out. We probably at this stage are fairly confident that this works. We probably don't need to keep doing that anymore. We're still doing it every now and again. It's easy. So we were approached, this is something we did recently, we were approached by Ambius, who is in the UK, so you're probably aware of the company Ambius. Big indoor plant hire company, and they wanted something done with one of their static systems, a little green wall in the chambers there on the left. We thought, well, this has been done to death. There's so many pollutant in chamber stages, let's do something novel. So we tried petrol, because petrol's a complex mixture of volatile organics. So it was interesting, it was novel. Let's see what happens when we try a complex mixture of chemicals and see if there's any preferential removal. And as it turns out, naturally, the plants removed the petrol vapor because plants remove volatile organic compounds. But the fun thing that happened in this project was the plants systematically removed the most carcinogenic volatile organics first. I mean, through what coincidence that occurred is neither here nor there, but that's the way it worked. The aromatics were removed more efficiently than the alkanes, which were removed more efficiently than the oxygenated hydrocarbons, and so on. So that made a very good paper, and it made very good media. And we got well over 250 million views for the media associated with that project, which I think, I mean, not saying it was a stroke of genius, it was the plants did the genius, not us, but that is a good indication of exactly the impact of the work that we're doing here. If you can show that plants have a meaningful effect on indoor air quality, people are fascinated by it and they love it. A lot of the reason for this big media impact was that it got picked up in India. And of course, in India, they've got significant indoor air quality problems, and it was very high impact project. Plants remove all sorts of air pollutants. I'm not going to talk about mechanisms. I'll leave that for the chemists. Shervin. Ozone's removed. It kills the plant slowly, so the plant functionally removes ozone from the air. If you expose them to too much ozone, the plant will die eventually. Oxides of nitrogen removed, oxides of sulfur removed. It's all quite effective. In indoors, the big pollutant, particulate matter, is more challenging. And basically, a lot of what I'm saying is leading into Peter's talk. Particulate matter is by far the most important air pollutant. It kills 2/3 of a million people a year in China. It's very dangerous. For plants, their natural plant surfaces are quite adhesive. So particulate matter in the air does stick to plant surfaces and is adhered, but that removal is temporary. You can take plant leaves that have been in a polluted environment and wash them, and you can see the particulate matter's accumulated. But in situ, most of that retention's temporary. It's liable to be resuspended under water and under wind. Most importantly, if you have an environment, an urban environment, particularly an indoor environment that's particulate polluted, it's very hard to detect the effect of plants on that air quality. So we'd have to conclude that particulate matter removal by indoor plants is probably trivial. The effect sizes are probably too small to make any sort of difference. But don't keep doing it. Recently, we delved into carbon dioxide. One of our favourites. There's quite a few papers out there on carbon dioxide removal by plants. The mechanism is rather obvious. Plants what am I saying? Plants photosynthesize and they remove carbon dioxide. It's what they do. The point here is that in the built environment, 30% of all the energy used is through ventilation, the purpose of which is to flush out carbon dioxide exhaled by people. So if we're ever going to get to net zero, and the International Energy Agency and the World Health Organization are very clear on this, we need to reduce energy demand. So in the urban environment, The biggest user of energy is ventilation, removing carbon dioxide, and plants do it for free. There's clearly a concept here that's worth pursuing. This could be a very major thing. So, like the picture on this slide, the graph, lots of studies put plants in chambers, shine light on the plants and measure the carbon dioxide going down. Some species are better at it than others. Palms are particularly good at it. Here we are in this particular experiment, which we had published, the date's wrong, it's 2026, we had published this year. We tried a crop species, we tried bok choy, and naturally being a fast growing food crop, it photosynthesized rapidly and pulled down carbon dioxide very quickly. The rest of these are ornamental species that you know. So really good effects. If you go and if you can do in situ studies and you have a sealed room and you put enough plants on it and shine enough light on the plants, you can maintain the carbon dioxide in that room very effectively. So the effect size is room scale. Only one problem, of course. In indoors, artificial lights are typically used on plants, and artificial lights require energy. So the question we asked here, and this is a question that's been wearing at us for over a decade, can we actually make ventilation equivalents with indoor plants? So we put plants in chambers, measured the amount of photons the plants were absorbing, and related that back to how much carbon dioxide they were pulling down. These are the graphs. The most effective plant we had was the bok choy. It pulled down about 4,500 milligrams of carbon dioxide per kilowatt hour through the light energy, which sounds like a lot, 5 grams per kilowatt hour, until you compare it to mechanical systems, which are up around 170,000 milligrams per kilowatt hour. So I think we reached the end of this continuum with this study. You're not going to have indoor plants that are artificially lit that are going to have a significant and valuable effect on ventilation. They use too much energy. The lights use more energy than you would expect. However, going forward, and this, I think our role as scientists and engineers in this particular research trajectory is going to end here, because the next stage over to the architects to design buildings where we can have naturally lit plant-based systems, which then could plausibly replace ventilation. Then we're making real international broad-scale impacts and helping that move towards net zero. That is a big ticket item. Yes, I am giving away research ideas. Please use them. We can't. They're too hard for us. These are free. Add me as an author if you like. That'd be my pleasure. Something else that's interesting, that's kind of new, is the idea of negative air ions. Negative air ions are produced by plants amongst some other natural phenomena like rain. What they are is obviously negatively charged oxygen atoms. Their role, or their functional role, is to remove a lot of pollutants from the air, in particular particulate matter. So they cause particulate matter to precipitate. There is the most detectable difference caused by negative air ions is the difference between air in a natural system and an urban system. You know, when you go out to a forest, the air feels different. It feels fresh and clean. A lot of that affect's due to negative air ions. So what people have done said that might be a good thing. Let's test the effect of high levels of negative air ions on people. And there are a range of papers that demonstrate positive health effects from negative air ions. There's proven effects on human health that it improves them. And plants produce them. So our next question was, what sort of levels are plants producing indoors, and can that have valuable human health effects? So we did a study on that. A bit of a difference between indoor plant species, more leaf area, more negative air ions. They like a bit of light. Irrigation, negative air ion releases associated with water balance, so not surprisingly, irrigation had an effect. At the end of this experiment, we've concluded that in a decent number of indoor plants can produce significant and negative air ions to affect human health. So there can be positive health effects here. What needs to be done next is to go and measure that, is to get some of our health colleagues on board to go and measure the human health effects of these negative air ions. All good. So does this work in buildings? this is one of the big questions that I'd like to bring up. We did this study 2 decades ago. 120 university offices, no plants, 3 or 6, and we got a 50% reduction in total volatile organics. In fact, it brought the air quality indoors equivalent to outdoors. With the proviso, this study was done 20 years ago. This building was naturally ventilated. It did not have a mechanical ventilation system. So it's not representative of current state of affairs. So where we got up to then with potted plants is that they have a problem. And at the end of the day, the rate of pollutant removal is always going to be rate limited. For a plant to remove an air pollutant, the pollutant has to move through the air to the substrate, or the leaves, as the case might be, if it's formaldehyde, to get removed. So the pollutants have to move across with a concentration gradient to actually get removed, and that's slow. So indoor plants, passive potted plants, standard green walls will always be slow at removing pollutants. And the net effect of that is that pollution removal effects by passive vegetation are quite low per unit of green space, which means you need a lot of plants to have an effect. I think this is if the audience aren't familiar with this paper by Kumar and Warings, you should read this. If indoor air pollution and plants is of any interest to you, you need to read this paper. Cummins is quite a good engineer, rather Waring's quite a good engineer. He did quite a good study on this and worked and put plants removing pollution, not in our biological science terms, but in engineering terms, and worked out the clean air delivery rate produced by each plant for a range of pollutants based on previous literature. And the answer was not much. So an individual plant, like formaldehyde was the best pollutant from the literature, it'll produce 0.22 cubic meters of formaldehyde-free air per hour per plant, which isn't very much. And for other pollutants, all the way down to whatever that is for chloroform, virtually 0. So Waring published that paper saying that plants don't really clean indoor air and got a lot of press for it and made our job very difficult. And not without reason. Those numbers are quite small. So there is a challenge here that if we, as a field, are going to make a positive impact on the urban environment, we have to overcome. Sure, you can make plants work better if you use the right lighting. There's papers. If you use the right plants for specific pollutants in specific environments. If you choose the plants that work a little bit better, if you use substrates that support the bacteria that remove pollutants. There's even a paper from Zhang in 2018 on GM plants that were slightly better at removing pollutants. The individual plant as a pollutant remover can be improved, but at the end of the day, it's always going to come down to that isn't going to be enough. If in a modern building that has indoor plants, typically that is what they tend to look like, and that the standard potted plants isn't going to work, which is where that comes in, which is where Peter takes over at the end and talks about where we went since these realizations about the potted plant environment. I sound very negative. I'm getting old. It's not a negative story. All right, so wrapping up, 20 minutes on time. There are gaps, and those gaps in knowledge I'd like to share with you based on our understanding and our 100-odd papers in the field. What's next? Biogenic volatile organics are emitted by plants. They can act as air pollutants. There are people in the built environment who are rapidly communicating that indoor plants are not good for air quality because they produce volatile organics. These volatile organics can enter a reaction cycle with oxides of nitrogen and ozone to produce secondary organic aerosols. This research has not been done. That is something important. If someone can show that plants either do or don't influence secondary organic aerosol production indoors, that would make a major impact. Probably the most important gap we have, though, is how do plants actually perform in buildings that have people in them, opening and closing doors, opening and closing windows, running printers with computers, with ventilation systems with air change rates. That is very, very poorly studied, and that's critical to try and move our findings from chambers to the real world. All in situ empirical studies are priceless. if anyone out there is listening and who can do this work, plants should pull down viruses and bacteria. Plants produce negative air ions. They remove viruses from the air. It's plausible that viruses could pull down influenza and COVID-19. Hasn't been done and we can't do it. It's PC3 research, we can't do it. Finally, the next one that's a factor that's growing and growing in our experience is it worth it? Is it worth it for a company to go and spend significant amounts of money on indoor plants? How is that going to relate to their ESG targets? What is the carbon balance over the life cycle of an indoor plant installation? So life cycle analysis, engaging with our economist colleagues, very high impact research. And finally, the barriers to the use of indoor plants for functional purposes indoors are the same as always. Designing systems that don't take up too much space. With most of the systems, as I said, you need so many plants to have a real effect. Designing systems that will fit a lot of plants indoors. We still have our people who don't like us, who say we're just greenwashing. Why are we putting plants in buildings? It's just a greenwash. It just looks nice and it's a hippie science thing. Those people will always be there and they're easily dealt with data, with hard quantitative data. You'll always overcome those arguments, but we need always the data. And finally, back to the life cycle analysis, we get significant resistance based on return on investment. If people are going to spend a lot of money on indoor plants, what's the return on that? Can we do a life cycle analysis and do an ROI analysis at some juncture and work those numbers out? That, once again, will be priceless for our whole field and the industry as well. To finish, partners. Science is, I know Peter's going to show one of these as well. All we do is measure things in a lab or in buildings and we publish papers. It's our partners who put plants in and on buildings. So always value your commercial partners. They are the reason that anything happens. We've got universities, councils, private industry, China, USA, UK, all around the world. Thank you. These are the people that make things happen. And there we go. That's my vision of the future, which Peter can now jump in and pull to the ground. It's after your bedtime, Peter. Are you sure you're going to be able to do this? Thanks, everybody. Hope I wasn't too boring.
That was a whirlwind.
Yeah, thank you very much,
References.
Professor Fraser. I think it was fascinating presentation and to hear the insights on on your experience.
I just hope it's useful for you, for someone somewhere, because we are not that big a team. We've got 15 members. We can't do everything, and you're big and powerful. You can do this stuff.
Yeah. So let me then formally introduce our next speaker. So Dr. Peter Irga is a leading researcher in innovative nature-based biological air treatment technologies with a focus on countering the growth challenges of air pollution in urban environments. As a major coordinator for postgraduate environmental engineering and water engineering at the University of Technology, Sydney's Faculty of Engineering and Information Technology, Dr. Irga has made major contributions to the development of broad scope air cleaning systems based on green walls, known as botanical biofilters, advancing them to the point of becoming an emergent technology with multiple projects worldwide. His contribution to the field have been recognized with the 2025 World Green Infrastructure Network Award for academic work. Yeah, brilliant, and congratulations for this this award, Peter. So yeah, over to you.
Thanks for the introduction, Prashant, and Yeah, I'm not sure if I'm going to match the energy or the charisma of Fraser Torpy, but you know, a lot of us come into academia thinking it's a good spot for an introvert to succeed, and clearly some charisma is needed to communicate some ideas. So I've been had the pleasure of working with Fraser for my almost my entire research career now, and hopefully I've picked up some of those points. Which is excellent. Fraser is the science part of this story, and I like to think of myself as the engineering. When we're like, well, let's try to maximize the potential here of these plants that I didn't think were that limited, to be honest. Fraser kept saying, "Oh, they're limited in some respects," and I guess it depends on the air pollutant you're targeting. But I think if you still have potted plants indoors that are strategically placed that maximize the most amount of airflow past them, or in an atrium that can maximize airflow in a way that the air could be pulled into the building, that can still work. So that's where I think passive systems can really thrive. Otherwise, I've worked extensively on active systems, or our team at UTS has worked extensively on active systems, and so I'm going to talk to you about today. So with a passive system, you can see here, we've got a pot or a plant inside a chamber. And how we were used to do those tests, we put the plant inside the chamber. As Fraser mentioned, we'd fill up the chamber with an air pollutant and kind of monitor those pollutants through time. As you've just heard, we think that the area of the plant that was best at removing volatile organic compounds was actually in the substrate, in the rhizosphere. The bacteria on the roots were breaking down those VOCs after they were getting absorbed into the substrate, or sort like that. So how can we maximize the rate in which we can remove those pollutants? Well, let's put some engineering to it. So let's pull the air through the substrate. And the other, so we've got this active system. And one thing I'd like this GREENIN initiative to establish as well is some standard terminology that we're using here. So I'm talking active system here, and you can see the plants are vertically aligned. Now I would like to call it active green wall or active living wall. And there's another term we use as botanical biofilter. So I would really like some consistency across the terminologies here. And we're partially at fault here by using all these different terms interchangeably. We probably should have stayed with it. But as you'll see through the talk, some of the technology is vertical, some of it isn't. So maybe that's a bridge we can all cross together. Preliminarily, we developed a bit of a prototype here with a company called Junglefy, and we did a lot of work with Junglefy. As you can see, the system is vertical, it's modular as well. We would have, in the modular situation, we would have 16 plants. We'd pull air through the base of the system and then up through a plenum and out. And the idea, you'd have clean air coming out through the system. We've done a lot of R&D on that modular type system, mostly on substrate development. We really did not want any organic material to be in the substrate. We wanted to be inorganic because we had concerns of bioaerosols. So we didn't want to aerosolize any bacteria or fungi by flowing air through. So the substrate in itself is mostly inorganic. Yet we still need to develop it in a way that we can have both pollutant removal as well as water retention, as well as microbial colonization for just the rhizosphere. We still want gas diffusion. We still need nutrient availability for the plants as well. Speaking of plants, plant species is a very important thing. So a lot of R&D on what plants are going to work best. Even growing vertically is a challenge for a lot of plants. Are they going to grow well in low light conditions if they're going to be in the indoors? Obviously, their root their root system and how the roots are orientated within the module is very important for airflow. Airflow, a lot of R&D on that too, because if you can't just blast air through the substrate, plant's not going to really enjoy that. In fact, in some of the early experiments we turned the airflow right up, and we had plants basically getting thrown out of the module itself. And that led us to a situation where we tried to design a plenum behind the module such that the air flowed evenly and equally distributed across not only the module itself, but also multiple modules, and we could have multiple plenums there. And then thoughts around how you're going to irrigate these systems at such a rate. And the irrigation does have a role in pollutant removal down the track as well. So a lot of optimization, a lot of engineering. The early experiments were using a closed seal chamber, and we'd inject, there's a younger myself doing some of those experiments, injecting these VOCs into these sealed chambers. Fraser talking to some, hopefully, investors. What would happen there is the VOC would go into the system, and it would go through the substrate, through the plants. It will get passed through, get filtered through. But because it's in a sealed chamber, what's happening is that VOC is getting cycled through very rapidly within the chamber. In fact, compared to the potted plant experiments where it would take hours to days to remove VOCs, we could see VOCs removed within seconds. Honestly, within seconds. If I was to inject a hydrophilic VOC like formaldehyde, I would see it removed within seconds in a sealed chamber. And that came to some criticism within the HVAC world. Well, let's start comparing our experiments to what they do with HVAC filter testing. So we developed a flow-through chamber where you would generate a pollutant on one side of the filter, measure it, and then measure the filtered air on the other side of the filter, compare, and then you would come up with a term called single pass removal efficiency. Basically a percentage of the pollutant removed either side of the filter. And once we got this setup established, it was really Pandora's box. Here you can see I've got benzene in the chamber getting generated, but we could put a whole host of VOCs through this single pass removal efficiency flow through chamber. And we did. You can see acetone, benzene, cyclohexene, ethanol, all sorts of VOCs with all not consistent results. Different VOCs got removed differently, and also different plants had different efficiency as well. But for the most part, extremely good results. These results with this system were comparable to any VOC filter that you could buy off the shelf, to be honest. Which is really exciting. Now we have a real functional air filter that is botanical based. Of course, the take-home message here is different VOCs behave differently within the system. The more hydrophilic the VOC was, the better it was at getting removed. The more hydrophobic VOCs, BTEX, for example, they had lower removal efficiencies, and I think it's because of the moisture content within the substrate, which was required to keep the plants alive. Well, one of the target VOCs I thought we could start working with, cigarette smoke. What a perfect opportunity to start something crazy. So let's change that benzene to a cigarette, just hovering inside the chamber there. Let's start doing some cigarette smoke experiments. And we got some amazing results. This is with GCMS. For the most part, 43% of total volatile organic compounds, so average across all of VOCs, were removed. Interestingly, the best results we had was for nicotine. So that gave us some real exciting ideas around implementing this kind of technology inside an indoor space where you had a lot of smoking, or environments like that. I think maybe you can correct me on this, Prashant, but I think smoking indoors is banned in the UK. I think it is, but it definitely is in Australia. So maybe that's not our target market, but where you can, well, maybe we can start smoking inside if we all have a botanical biofilter indoors. Maybe that's our utopia we're all searching for. But you can see substantial pollutant removal across so many VOCs there. Of course, after we've put some cigarette smoke through, well, we've also got particles present. And that was a really interesting research direction that we could take this in. Particle removal. We didn't think it was going to be capable of such removal, but here you can see with total particulate matter, we're usually looking at about 50% removal efficiency. That is not as good as a HVAC filter. I will admit that. But the fact that this system could remove volatile organic compounds as well as particulates is incredibly exciting, because most conventional HVAC filters do not remove volatile organic compounds whatsoever, or any gaseous air pollutants for that matter. Interestingly, and you can see with the various root configurations from various plant species here, the denser and more fibrous the root is, the better it was able at removing particulates. So our best performing plants here, Nephrolepis exaltata bostoniensis, which I believe is Boston fern, is the most efficient at removing particles in this instance. 50%, that's astounding. Just like to talk about some research coming that we did in collaboration with the University of Antwerp. Because it's using very similar modular design, we were able to do some experiments where we were looking at optimizing airflow whilst balancing acclimatization of the plant microbe synergy for VOC removal, and we've got some really interesting results there. The plants can actually bounce back after being exposed to that VOC after a waiting period. They actually bring those same bacteria that were able to remove the VOCs back after a period of time, and this is using the exact same modular design that we originally designed. And subsequently, we also did some computational fluid dynamic modeling to enhance the idea around airflow, reducing pressure drop, and identifying dead zones within the system. So aerodynamic optimization. And I think using this research, we can really kind of take the technology from just a plant to now a fully air filtering technology that we're hoping to legitimize more broadly across the world.
Sorry, Peter, to interrupt. You have just 1 minute left, if possible.
Oh,
thank you.
Okay, no problem.
Thank you.
I didn't realize I was at such pace. And some other work in Spain utilizing this is with the University de Valladolid, looking at some interesting volatile organic compounds, especially from solvent cement, which is somewhat toxic. So that produces tetrahydrofuran, which is a neurotoxin. We're able to remove that as well. And I'll just quickly talk about some of the in situ applications that we had here. We're able to implement this in some of the buildings in beautiful Sydney here. And you can see the modular design leads to scalability here, which is incredibly important. Here you can see we've implemented it within a foyer within one of the tall buildings we have in Australia. And we're able to compare two different levels. One level of the building that had the active green wall and another level that didn't. Beautiful, to be honest. And you can see the effects here. So this is an in situ installation, not lab work anymore. This is in situ. Decreases in carbon dioxide, decreases in nitrogen dioxide, decreases in volatile organic compounds, decreases in particles. So that's in situ results right now. And we also got to test this technology in China, which was really exciting for us within a school. Prototype here, as you can see. One of the really exciting things is they had the top of the line HVAC filter, as you would expect with a very important school within China. And the botanical biofilter, in fact, outperformed it in terms of particulate matter and as well as volatile organic compounds. And I'll just cycle through some presentations here. So if you wanted a standalone system, you could utilize something like this, where you have a cylindrical system using the same modular system. And we tested that in situ as well. Substantial volatile organic compound removal in a much larger chamber now. And then we took that to an environment where we thought we would have the most impact. And that was at a train station that was under construction, that still was utilized as a train line. And now we're actually able to observe nitrogen dioxide removal, ozone removal, and particulate matter removal relative to a control office. So that's a new application that is indoors, but not necessarily a work environment like an office. It's an environment that people could literally utilize in terms of construction and transport. I might leave it there. I do have some more, much more, in fact. I think we could talk to another 30 minutes, in fact. Yeah, I suppose you'll have to join the conference circuit, and we can talk a little bit more at length on some of the work that's in this space. Thank you very much.
Yeah, thank you very much, Peter. I think we should get you back. The webinar series will continue, so
feel
maybe
free.
We could have a little bit more.
It's always a pleasure to talk to this technology.
Yeah, it's fascinating to see how the laboratory scale work could really translate into the real world sort of, the thinking, which is a kind of sometimes a major barrier. So you have done a fantastic job in there. So I think we got a few minutes actually to take some questions. So I will start with the one which Kalani Miller has asked. This question is for both of you, basically, so you can pick up any of you. Which talks about how much would the green walls cost if they were installed in older buildings like the ones in Sydney, and could they even be installed in most buildings? Also, how that formed aerosols impact the indoor climate other than cooling? Would it be an increase in aerosols also make them a harmful pollutant in badly ventilated buildings?
I typed an answer, and do you want to have a go? See if you give a different answer, Peter.
Oh, well, let's explore your answer.
I said the upfront costs are currently high because that's a niche product, but with economy of scale, if more people were using these systems, the costs had come down. There's nothing particularly expensive about the systems. What was next? So
the cost will be... You can't really put a price on health, I heard a wise man once say. So if you're able to improve anyone's health, that should pay for itself, in effect.
Someone needs to do a return on investment study on this. Just saying.
Yeah.
And as you've probably heard through all the talks leading up to this one, all the psychological benefits, all the well-being benefits of having plants present, and I'm not saying that they're not. They definitely do cost, like everything does. But there was a period of time where electric vehicles were unattainable for the common man, and now they're the most highest selling car that you can find because there was economies of scale. There are more manufacturers of them out there. It's not a niche product anymore. So I think that's the direction that we're going.
Okay, perfect. Yeah, so I've got some more very interesting questions. So this one is from Bala Good, and they're asking, when you use edible plants like bok choy, are the levels of contaminants too high that they are not edible, or are contaminants only in the root system as it absorbs the most?
Great question. That's a good question. And the answer to that is we don't know. We have done some research on vegetable crops in outdoors, and we found significant uptake of heavy metals. So we planted vegetables in an outdoor area in Sydney, which is pretty polluted. And our first crop was highly contaminated, and it wasn't suitable for human consumption because of urban air pollution. So we had to throw it away. But the second crop was fine. So what it looks like is that the first crop is going to functionally mitigate any residual pollutants that are floating around, and the second crop will be then safe. I should also point out that we tested supermarket food crops as a reference, if you like, to our outdoor grown vegetables, and they were also high. But in Australia, we don't test for them. So yeah, anyway.
With respect, that was with particulates, and a component of those particulates were heavy metals. Most volatile organic compounds will get broken down to its basic constituents, which would be a basic hydrocarbon, which is non-toxic, and water vapor, and plant material. So if you're just putting volatile organic compounds through a plant system, I think you can possibly eat those crops. It was just heavy metals that don't necessarily get broken down. They get bioaccumulated by the plant.
Yeah, that was my next point.
Oh. Yeah, so I got a question for you, Peter. So I saw that your comparison between the active and the passive sort of, or where there was no green wall in the real in situ sort of thing. So I also saw that the fungal pores actually they increased. When you put the active wall, so there is a pollutant removal, so they all got decreased, but the fungal spores increased actually. So what do you think was the potential implications of it, or do they outweigh actually the benefits you get?
Sorry, Prashant, is that with humidity?
No, so the last slide actually you had where you were showing the comparison between the active, the system, and I think it was in Sydney, and then you compared with a place where there's nothing there. So all the VOCs, PM10, they all increased, but the fungal spores increased actually in the table.
The fungal spores did increase, yes.
Yeah.
So I'll just quickly go back to that slide. That was an important part that, and it really fascinating that you picked that up there. Yes. There was a slight increase. I think that's to be expected when you bring that volume of plant material into a indoor environment. But the World Health Organization cutoff here is 500 colony forming units per meters cubed. So we're well below it, even with this volume of plants. We've also seen in other situations where fungal spores have actually been filtered out by the systems in a similar way to particles because they're going and getting trapped within the substrate itself in the same way PM10 would. Interesting observation. I did kind of glance over that, but anything below 500 is still deemed a safe environment. I don't mind getting proven wrong of that. Or you can determine the indoor-outdoor ratio when it comes to colony forming units with respect to fungal spores. In this instance, we did not. But I can tell you in a tropical or sub-temperate kind of climate that we have here in Australia, most outdoor fungal concentrations are incredibly high.
Yeah, so I think we got a number of kind of, the questions. We never had these many questions before, so which is a great thing. Maybe we just take one or two more because we're already, the end of our time. So maybe I'll just pick up some random ones basically there. So I'll pick one from Richard Sebin, and he asked, building regulations in the UK require mechanical ventilation. How and when could we get the stage of convincing building regulation officers that can generate fresh air in a space with plants and turn the mechanical ventilation off. Because currently, the air that we improve is sucked out by the mechanical vents, and fresh air is drawn in from outside, making it more challenging to show the impact of activity in walls in commercial environments.
That's where I was going, I think, with the in-situ study. We need more compelling empirical in-situ data that shows that you can functionally turn off the HVAC, the ventilation, and still get good effects. Currently, that data doesn't exist. We've got this proof of concept, which is good. It's strong. Our numbers are good. We know it's going to work, but that in-situ data in a mechanically ventilated building isn't there yet. We don't have a basis for making that argument. We can't go down that road yet. We need to.
Yeah. Okay, great. So I'll pick up one last question. So this is from Jill Thompson. And they ask, for the green wall, did you try taking the air through the front of the wall and collecting the water transpired or water vapor and air and filtering it before pushing it back into the room? Could maybe use organic and more plant-friendly substrate. So it's basically where the air is going in and where it's coming out,
So,
and whether this passing through the substrate.
I think that would work really well, and it does work well in a standalone system that isn't a green wall situation where there's ducting and things like that. If you've got a in-room kind of column style botanical biofilter like I showed, I think that would work, but at scale where there's ducting involved, I would have concerns that there's potential mold build-up in the ducting, depending on the airflow.
Perfect. And if I can ask a very quick question that is coming from Satoris, he asked, what is the best soil mixture?
That's a tough question, isn't it? But all our stuff's based on coconut husk because it doesn't produce mold spores and it doesn't support the growth of Legionella. So we'd go high lignin, high organic substrates. So hemp, linen, maybe flax, that sort of substrates seem to work best if you want to, especially if you want to use airflow. That's important because we can't have Legionella. What Peter didn't mention is when we tested the fungal spores, we also tested Legionella and it was fine. If you use composted organic material that composts readily, you're going to risk that Legionella. So we say high organic, high lignin, and testing. Test the substrates if you want to put air through them. Does that answer the question?
Perfect. Yeah, thank you very much.
Thanks.
So can we give a big round of applause to our fantastic speakers today? It's been a real pleasure listening to both of you. And we take this an opportunity to announce actually our next webinar that will be on 1st of July, the same time on Wednesday. And we got another 2 brilliant speakers. So one will be our own Tiana Blanchua. So she has been also very closely associated with the Green Network, and you know that she comes from horticulture, Royal Horticulture Society, and she has done a fantastic job actually in this particular area. So you can hear more about the plants and where are the the devil in the detail. That's a really interesting title, so I really like it. So yeah, we'll welcome you to the next webinar, and then the second speaker we have a early career researcher from University of Liverpool, Dr. Bethan Mead, and she will be talking about the urban agriculture, diet, and public health. Wow, that's another very complementary and interesting topic. So yeah, we are bringing you the best of the experts actually in this webinar series, and we would really like to have you back for the next webinar there. But before you head off, so just wanted to say a few things. That you can become a member of the GREENIN network for free. So there is a barcode there. There's also the link, but you can go onto the GREENIN website, and then you can see a tab actually we say join us. So it's free. It just takes like a few minutes to complete the form, and you will start getting all these the information on the webinars and other activities there. And if you are on the social media, especially on Blue Sky or LinkedIn or on X, so you can see our handles for the GREENIN. So please do follow us. That will allow us, you to remain updated with the day-to-day activities of the network and the fascinating, the work the network colleagues have been doing. The one more thing I would like to say to everyone, we send our monthly newsletter every month that goes at the end of the month. So if you have any fascinating research you published or any projects you have been working on or any opportunities in this particular area, please do feel free to send an email to us. So we will be very happy to include in the newsletter and promote that activity for you. So this is an open invitation. And if any of you wants to be a speaker on the webinar series, we will really welcome you. So please do send us an email. So with that, again, the huge thanks to our speakers and coming from Australia and taking your time off from the evening, and thanks to our team as well. Kanika and Sarvan for helping with the organization, and all of you for coming and listening to our fantastic speaker. So this will be on the YouTube very soon. You will receive a link so you can have a look in more detail. So Peter, you are most welcome actually to get all your slides in if you wanted to, but otherwise we'll possibly ask you to come back at some point and go through the rest of some of the interesting work which we couldn't see today. Yeah, thank you very much, everyone.
Thanks so much for having
Thank you.
us. Love you all.
Thank you, Peter and Fraser. Thank you very much, and have a good night there. Bye.
Thanks for having us. Back to convict work. Let's go, Peter.
Because we're in Australia.
Yeah.